easiest time because you are just after pretty good lunch, and most of what you want to do now is another talk in mathematics. But I know you by now. But that's not going to happen. And instead, this afternoon is mainly for really cultural tour that starts by a lecture by our really honorary guest of Benny Shilo. Benny Shilo came from, is a professor at Weizmann at the Weizmann Institute of Science in the genetic departments, and I think I know you for more than I would like to even remember, but tens of years, mainly because I think I learned quite a lot from Benny on development, on communication between cells, and even now, this is one of the most interesting topic that I, in my lab as a biologist, are interested in. So, but now you are going to have a different type of tour in science and art, but also around development, I guess. And I won't say much more, but two things of organization. I'm not Nati, if you are confused, but he apologized that he couldn't be here because all those recruitment committees now. But immediately after the talk, at about 3, we are going, and who is leading us? Okay, so just follow. So you will be walking together to the museum, and there will be a guided tour there. And I think the other announcement will come later, but just not to take too much of the time, let's thank Benny and enjoy the talk.
Hi, it's a real pleasure to be here. I really like this place. I organized the developmental biology school here almost 20 years ago, which was a wonderful experience, and it's nice to see that you're experiencing the same magic atmosphere here. So I saw the list of topics in your course. I couldn't understand the word. Hopefully what I'll say today will be more legible to everyone. So I'll talk about embryonic development and essentially about one of the biggest puzzles in biology and in life. How does the structure of the embryo form, not just in humans, but in all species? How is it so precise? How is it so reproducible? And where are the rules that guide this magic patterning process? And we know that all the information, everything begins upon fertilization. This is not the landing on the moon, it's the sperm fertilizing the egg. And essentially, the DNA which is contained within the sperm and within the egg will merge together, and this single cell, the genetic information within this single primary cell, will essentially be the information that all cells of the embryo and later on the adult body carry with them. And somehow this information conveys the instructions on how to build the entire body plan. I should also say, if you have burning questions in the course of the talk, don't hesitate to ask them. So why do we know that the instructions are embedded within the DNA? Because if you look, for example, at identical twins, they are indeed so similar, not necessarily identical in pattern. These are already epigenetic changes, but they are so similar because they've received the same genetic information, the same instructions to form the body pattern.
What does it mean epigenetic?
What does it mean? Maybe we'll talk about some of the less burning questions and deeper ones. Let's postpone them to the end, but keep the question in mind, okay? So the whole field of developmental biology and our understanding of how embryos develop has undergone a major revolution in the last 30 years or so. Most of the public is not aware of this revolution, and it concerns each and every one of us, both from the curiosity aspects and from medical aspects, and therefore I think it's really important to convey all of these new developments to the broad public. So this is the science that's behind the embryonic development that has really developed. And I was thinking of a way to combine or to convey this new information to the public in a way which will be readily accessible to a broad range of people with different backgrounds and so on. And I decided to convey the science to the public by combining the recent scientific findings with photography, with my love for photography in a way which I'll describe in a minute, and somehow by this convergence maybe achieve a more effective way of popularizing science. And actually did it 3 years ago at the sister institute to this institute, at the Radcliffe Institute for Advanced Studies. And even though I'm a scientist, I came there under the auspices of or under the pretense of being a photographer or an artist. So they gave me a huge studio which I filled over the year with pictures and so on. It was really a very memorable experience. So what's the idea? The idea is that there's a saying that a picture is worth 1,000 words. And the reason is that when you look at a picture from the human world, it immediately conveys memories and recollections and so on. Each one may interpret the same picture in a different way, but it comes loaded with a lot of meaning because of all of your previous histories and experiences and so on. On the other hand, we generate scientific pictures which are very useful, very important, very informative. They have a lot of background in them, but the general public, of course, is unaware of all that background. And therefore, I mean, one can actually say that a scientific picture is worthless without 1,000 words. And you can look at this beautiful picture of the muscles of a fruit fly embryo, and the baby is the only one who is looking at the pictures. The parents couldn't care less. And for them, you know, it's just a bunch of nice colors and patterns, but it really doesn't mean much. And the question is whether you can combine these two aspects in order to convey science in a more effective way by generating or by using the human world and human introduction, human experience and so on, in order to generate paradigms for scientific rules and understandings that will be readily amenable to the broad public. And this is what I'm actually trying to do in this project, to combine these two kinds of pictures in a meaningful way, as you'll see, which is not necessarily a visual representation of scientific aspects, or I mean a visual citation, but more of a conceptual paradigm for scientific concepts that I'm trying to convey. So this project started as a photography project, and I've generated these pairs of images which were displayed as an exhibition in several places. Later it actually matured into a book which was recently published by Yale University Press, and actually there's a website which really allows you to download many of the pictures and the chapters and so on. So it's a very interactive thing. And there are the lectures, of course, which are another way of presenting the idea. So actually the idea of using the human world as a paradigm for biological principles is not new. And Robert Hooke, who was a very prolific scientist and architect and physicist, biologist, you name it, has built one of his first microscopes, even before the Leeuwenhoek microscope. And under this microscope, he looked at a section of a bark of a cork oak tree. And looking at that bark, and these are his drawings, he saw these compartments, which we now know are the boundaries of dead cells. And he said that these look like cells of monks, so we will call them cells. So in effect, the first definition of the word cell in a biological context came from the metaphor to the human world. And what I'm trying to do, as you'll see, I mean, there are many similarities between the behavior of cells within the embryo and the behavior of people within a human society. So essentially what I'm trying to do is to make the viewer feel like, or the reader feel like a cell within a developing embryo, and actually feel the concepts rather than just understand them in a much more direct manner. Okay, so the first question we can ask ourselves is, why are cells in our body different from each other? I told you that they all contain exactly the same genetic information. So if they all contain the same information, why do they behave in such different ways, such that a muscle cell that you see over here will have the typical morphology of a muscle cell, a nerve cell will look like a nerve cell, they have different proteins that they express, and so on. And the answer is that even though all the cells have exactly the same genetic information, actually each cell chooses to express different parts of that information. So in a way, it's like, this is by now, I guess, an old-fashioned analogy. Most of the younger people may not know what it means. This is a card catalog in a library. There used to be libraries once. And the catalog contains the information for all the books in the library, but you can choose which ones you want to look at depending upon your direction of interest. So the cells actually, depending upon their fate or tissue, choose which genes to express, and in this manner which proteins to express, and eventually this will determine their shape and function and so on. So the choice of what to express from the vast genetic information that you have is essentially what makes the difference between cells. But in a way, this is the end of the process. I mean, this is already a stage where the cells are differentiated and different from each other. We have a muscle cell, a nerve cell, and so on. What we really want to understand is how does this process of allocating different fates to different cells come about. I mean, we start with cells that are initially identical to each other, and eventually they have to generate an elaborate, or to orchestrate the formation of a very elaborate tissue, like, for example, this is the hand of a mouse embryo. So you can see the muscles in red and the nerves in blue and the tendons in green, and everything is very highly orchestrated and coordinated and so on. So how does an identical population of cells somehow form this elaborate and reproducible structure? It's really the process that we want to understand, not the end point of the process. And perhaps one of the seminal experiments in developmental biology which really set the way that we think about these processes today is the experiment of Hans Spemann and Hilde Mangold in 1924. And what they've done was the following. They took a frog embryo, and from an early frog embryo, they took a region of the embryo where the first fold appears, and this will be the region where the head will form. So they took that bunch of cells that will eventually form the head, and they transplanted it into the flank of another embryo. This is the donor tissue, and this is the recipient embryo, and into a region of the embryo that will normally form the flank of the embryo. And as you see, they've used two different types of embryos. The donor was albino, the recipient was pigmented, so you could tell which cells contributed to which structures eventually. And the amazing result they got was this kind of embryo, where instead of one head, we see a very precise duplication of the head structure. Essentially, you can't tell apart which is the original head and which is the secondary head that was formed. So you could say this perhaps is not surprising. I mean, you take the cells that will make the head, and indeed they made the head, but the surprise was that most of the cells that contributed to the formation of this head are actually cells that arose from the recipient embryo. So what this means is that the cells would normally develop in one direction, but as a result of encountering this cluster of cells, they've now changed their developmental fate and produced something else. So in other words, the cells are talking to each other. These cells somehow communicated with their neighbors and impinged upon the direction that these cells will follow. So he coined the term induction, which means that these cells induced the neighboring cells, they talk to them, and so on. So this is, I guess, the biggest surprise in this experiment. I mean, how cells can change their fate. There are two other interesting aspects here. One is that, as you will see in a minute in the movie, you perform such a crude manipulation, and yet you're able to generate such a precise structure. So how does a crude manipulation give rise to such a precise outcome? And the other question, which is interesting here, is that because the cell number is limited, here we're actually forming two embryos in the space of one, so there are less cells per each embryo. But if you follow different hallmarks, you'll see that the proportions of this smaller embryo are well maintained compared to the big embryo. So how do you maintain proportions in patterning? So just to show you a movie that will actually exemplify, or I should say the analogy that I thought about was for this experiment is the fact that in essence developmental biology is not about predetermined fates that cells follow, but it's about making decisions and communicating with the neighboring cells, and this communication affects the ultimate choices that you make. So it's really like continuous choices that cells have to make, and if we want to understand development, we have to understand what these choices are and how the cells communicate and how they go about making the right choices. And of course, I mean, looking at this picture, you cannot help but think about this wonderful poem of Robert Frost.
Sorry?
Yes?
Just a question for
Yeah.
Was similar experiment ever conducted on human beings?
No.
How different is the human embryo?
Good question. Let's again keep that towards the end of the talk. We'll discuss it quite a lot during the talk. Okay. I mean, one difference here is that the embryo develops outside the body of the mother, so it's easier to transplant and maintain it and so on. Okay, so just to show you a movie for this kind of experiment. So this experiment was reproduced by Eddie De Robertis, and so here he's taking the future head region from one embryo. As you can see, he's not a great surgeon. You wouldn't want him to operate on you. Very crude. You take this bunch of cells.
Are these two embryos genetically identical?
They could be, but it doesn't really matter here. There's no rejection, immunological rejection in this time period. And now he sort of makes a little kvetch, and the cells will intercalate into the other embryo, and after 36 hours you get this amazing result. So very crude manipulation and a very precise outcome in the end. So how does that happen? And this fantastic experiment was actually followed by 50 years of complete frustration, where people tried to isolate these magic factors that cells use to communicate with each other and couldn't do it for a variety of reasons. I mean, one is that this is a very complicated bioassay. The other is that these critical substances are present in minute amounts, so it's very difficult to isolate them. So really 50 years of complete frustration at a time where molecular biology has undergone a tremendous revolution in terms of deciphering the genetic code and a variety of exciting findings. But still, I mean, as you'll see, the solution came from a very different direction. So just to highlight the complexity that we are facing, embryonic development is complicated because unlike assembling an airplane or assembling a Lego structure, the parts are not prefabricated. So where do the parts come from to make the embryo? And who puts them together? Or in other words, who is the conductor that orchestrates the whole process? Again, unlike building an airplane, there's no main engineer that supervises all the different departments. Who takes care of coordinating all these activities? So the answer came from a different direction, and it started to emerge about 30 years ago. It came from the fruit fly. And fruit fly research is actually not a new invention. It started over 100 years ago by Thomas Hunt Morgan in Columbia University. You can see him here with his collection of fly bottles. And he actually isolated the first mutation in flies over 100 years ago, slightly over 100 years ago, the white mutation. And since then, the field has really revolutionized biology in a variety of directions. So what's special about flies? Well, one advantage that they have is that they have a short life cycle. So the time from the time that the female lays a fertilized egg until the embryo develops and forms a larva and a pupa and so on. This is about a 12-day cycle, so it's a short cycle. Every female can lay up to 200 eggs, so you can get many progeny. Embryonic development takes place outside the body of the mother, so it's easy to isolate embryos, synchronize them, and so on. So they're amenable to sophisticated analysis. And the combination of 100 years of genetics with molecular biology and microscopy and transgenic flies and so on really generated a very, very powerful field. Now, the approach that was employed to understand what are the rules that guide development was essentially a genetic one. So what is a genetic approach? The idea is that you approach such a complicated process like embryonic development without any preconceived notions about the process. You know nothing about it and you are very humble, you don't have any theory about it, it's too complicated for you to invent a theory, and essentially what you do is come up with a powerful assay to look for defects in the process, and I'll show you the assay in a minute. And the idea, in the fly we have 13,000 genes, so the idea is that if you mutate at random each one of these genes and then look at the deficiency in each one of the genes separately, then you may come up with the ones that, when absent, give rise to defects in the process that you're looking for. So in a way, I mean, you can think about genetic approaches as the presence of the absence, if you will. You remove something and you ask what goes wrong in its absence, and from this you infer what's the normal function of that gene. So I tried to convey this idea in this picture that was taken in a guest house in Hampi, where the leaves that were used here for the imprint are no longer there, obviously, but they have a very strong presence. So what approach was used in the flies? So fly embryos take 24 hours to develop. By the end of these 24 hours, they have 50,000 cells, and they emerge as larvae that crawl and eat the food and so on. And the external cells of the embryo, the skin, if you will, secrete a hard cutinous layer, which is called cuticle, which you see over here. It's essentially like an external skeleton. And this layer is like a skeleton, very resistant and resilient. You can boil it, you can do whatever you want, it will remain intact. So it means that even if the embryo dies, just like a skeleton or like a fossil, the outer envelope will remain intact and we can look at it. Now, luckily, the outer layer that is secreted has a pattern. You can see segments, you can see head structures and tail structures, belly structures, back structures, so it has a pattern which reflects the pattern of the cells that secreted it. So if there is any defect in the differentiation of the cells that secreted this pattern, it should be readily identifiable when we look at this outer skeleton. And this was indeed the case. So looking at a collection of 18,000 mutants, they could identify ones which are not just dead but have a normal pattern, but specifically ones that are dead in a way that is interesting to us. Namely, the pattern will somehow be altered. Maybe the head will be missing, maybe the tail will be duplicated. Hard to predict a priori, but something which will affect the pattern. And the results were astounding in the sense, and this experiment was done by Yanni Nustein and Eric Wischhaus, and they received the Nobel Prize for it in 1995. Spemann received the Nobel Prize in 1935. So there were a couple of big surprises that came out of this analysis. The first one was that even though the pattern of the embryo is quite complex and elaborate, and the fly genome has 13,000 genes, there were only 200 genes that were involved in generating this pattern, an extremely small number. Furthermore, you could identify embryos which showed a similar defect but were defective in different genes. So this again would indicate that maybe these genes work together, and when one is missing or the other is missing, you would get a defect. But again, it pointed that not only we have only 200 genes, but they actually can be clustered into 5 or 6 groups, and that's it. So what are these groups that were identified? Essentially, they represent the genes that make the proteins by which cells communicate with each other. So these are essentially the magic substances that Hans Spemann predicted. So how do cells talk to each other? Well, one cell that wants to send a message can make a protein that will be secreted out of the cell or maybe displayed on the membrane of this cell. And neighboring cells that will receive the message have on their surface proteins called receptors, which can interact with this hormone. And once they interact with the hormone, this will alter their shape, conformation, whatever, and will lead them to activate a cascade of proteins that will eventually culminate in messages to the nucleus. Everything has to culminate in the nucleus, because as we said in the beginning, it's the genes that you express, which obviously reside in the nucleus, which will determine what you are and in which direction you develop. So it's essentially a linear pathway that conveys the information from one cell to the other. And the reason that several mutants in different genes exhibited the same phenotype is that you can block the same pathway in different ways. You can eliminate the hormone, you can eliminate the receptor, you can eliminate the cytoplasmic factors. Since it's a linear pathway, they will all give rise to the same defect. Now, the third surprise, which is perhaps the most dramatic one, is the following. I mean, you may say, who is interested in learning about fly embryogenesis? This really has nothing to do with us. And maybe the principles and genes that guide the embryogenesis in the fruit fly are so remote from the ones that guide our own development that this is really just an academic curiosity. But as it turned out, these genes and pathways that guide communication between cells are so ancient evolutionarily that they were all present at the time that the first multicellular organism appeared on Earth about 600 to 800 million years ago. So essentially, these pathways embody the essence of multicellularity in all multicellular organisms. And therefore, these proteins and genes are conserved throughout evolution. You can find them in worms, in hydra, in humans, wherever you look, you'll find them in a very, very similar conformation because they arose from the same ancestry. And there are essentially only 5 or 6 of these communication pathways between cells. So if you will, they're all conserved evolutionarily. They have different names that are not important at this point. And you can essentially say that cells, in the context of embryonic development, speak only 5 or 6 languages. Each one of these languages is comprised of a single word. And with this limited number of languages, the cells have to generate these elaborate structures that we discussed in the beginning. Now, not surprisingly, and perhaps we'll get back to it at the end, since these genes are so central to all multicellular organisms, it turned out that a variety of diseases, cancer, other genetic defects, and so on, are all related to the components of these pathways. And in a way, biology became much more universal once these discoveries took place, because it suddenly became clear that you can study human cancer, not necessarily in human cancer cells, but you might as well study the eye of the fruit fly and learn more about that pathway than you would in a human cancer cell, because of this enormous conservation and universality. So, in a way, I mean, you can think about these signaling pathways as a playback improvisation theater. Here the actors are passing an imaginary object from one person to the next. The object changes its shape and size as it is transferred from one person to the next. So now that we know that cells communicate and we have the machinery by which they communicate, we need to understand how this communication brings about the formation of pattern. And you can think of other processes which have to do with pattern, with communication, and with following rules that will somehow maintain the pattern. One nice example is this flock of starlings. I mean, look at the way these birds are forming the cloud. I mean, obviously they are communicating with each other. There are some rules. They may not be very strict, but there are some rules that maintain this cloud together. And so it's about communication and rules that give rise to pattern. And the patterns that we want to understand are the patterns of the embryo. And let's start with a simple pattern. This is the outer layer of the skin of the fruit fly pupa. And it's lined with these red cells that are neuronal cells. These are neurons. And as you see, they're interspersed by cells that are non-neuronal. And it's a fairly regular arrangement of cells, fairly fixed distances between them. But it all started from a population of cells that were identical to each other. No one told this cell to become a neuron or this cell not to become a neuron. You start with an identical population of cells, and with a single rule and with a single communication pathway, you bring about this pattern. So how does it happen? One other thing that we did a lot at Radcliffe was drink a lot of coffee. So there were 3 flavors of coffee, so you'll see how that can be used also. So let's start here with an identical population of cells, and we want to reach this pattern of interspersed neurons. So the way that it works is that even though the cells are identical to each other, there are random fluctuations in the level of the hormone that each cell produces. And once, for a given cell, once these fluctuations reach above a certain threshold, that cell becomes a neuron, but in parallel, it tells its immediate neighbors not to become neurons. So we start with the situation where all cells are brown, then one cell becomes a neuron, and then it tells its neighbors not to become neurons, and then another cell will do the same, and so on and so forth, and you end up with a relatively reproducible pattern that you see over here. It's not 100% precise, but it's pretty good. In a way, like spacing of umbrellas on a beach at regular intervals. Let's see how this happens in real time. So we can actually mark the cells in real time with fluorescent proteins, and you'll see how the process takes place. Here is the region where the patterning takes place. And you see the red cells, the neuronal cells appearing within the green cells, and eventually you generate this reproducible pattern. So essentially the cells, there are random fluctuations, and of course the timing has to be very happen within discrete values in order to allow this to happen properly, and then the cells communicate the information to their neighbors. Now I mentioned earlier that we can infer the function of genes by their absence. So what happens if we make these cells deaf to their neighbors? They can talk, but they can't listen. This is a phenotype that is common among many people, not just among these cells. So what will happen? I mean, all these cells have the capacity to become neurons, right? So if they fail to receive the information from their neighbors, then they will all become neurons in the end. So here you can see this patch of cells. Within a normal population, there's this patch of cells that are missing the receptor. They are deaf to their neighbors, and therefore they all became neurons in the end. And from this we can infer what is the normal function of the receptor, what is the normal function of the pathway. In a way, this is more, in contrast to the previous picture, this is more like the spacing of umbrellas on the beach in Tel Aviv, where the people are more oblivious to the signals from their neighbors. Okay, so in a way, where the question has almost been reversed. So we have a very complicated process. We have a very small number of communication pathways and a few simple rules, and the question is reversed in the sense of having to account for this complexity by such a limited number of signaling pathways and their rules. So what I'll try to do now is to show you how we can use these pathways and combinations of the pathways in order to generate elaborate structures. So one rule that the cells follow is not to be faced with this situation. This is Davis Square in Somerville. You enter the intersection, you have 6 possibilities. You're bound to be lost. You can't make so many decisions at one point. So the idea is that cells have to make simple decisions, meaning that at every junction it's best to make a binary decision between 2 options, and you reach complexity not at once in a single decision, but rather by a succession of progressive decisions that build on top of each other, each decision being a relatively simple one. So in a way, I mean, this can be like a game of dominoes, where at every step of the game, it's a simple maneuver that you do by following simple rules, but by the end of the game, you form a pretty elaborate structure, which is a combination of the different steps that took place during the game. And the same is true for development. I mean, if we look at the fruit fly embryo, you start with a simple gradient of a protein, which is high in the future head and low in the future tail. This gives rise to expression of genes that subdivide the embryo into 3 domains, and then these genes talk to each other and subdivide the embryo into 7 domains, and so on and so forth. An accumulation of decisions that gives rise to a complex pattern. And implicit in this idea is the fact that there is a hierarchy in the decisions. The earlier the decision, the more important it is in determining the final body plan. And the first decisions are obviously the most important ones. I mean, if you go out of this gate and you have to decide whether to turn right or left, I mean, this is the most critical decision you make. Later on, it's sort of a refinement on the first decision. And in a way, this is one of the answers to the Spemann experiment. I mean, the reason that such a crude manipulation could give rise to such a precise duplication of the head is that what the experiment has done was to recapitulate the first crude step, something which is similar to this gradient. Everything else followed according to the normal rules that carry out the refinement of the process. So, I mean, just to give you a feeling for how the initial decisions are the most important ones and they lay the basis for everything else. I mean, what's this picture? Right. So, I mean, not much difference between the two, right? I mean, the first step was already the most critical one. Okay. Another important way in which these 5 different pathways can provide a variety of decisions and cell fates is the fact that these pathways do not necessarily work in isolation. You can actually combine pathways and generate a situation where only if 2 pathways are active at the same time, or only 3 pathways, or one pathway could be activating, the other one could be inhibiting. So by combining different pathways, you can generate much more refined outcomes. I mean, this is in a way like a palette of an artist, where you start with your basic colors, but you can generate an infinite array of combinations. And just to give an example here, so here is the gene expressed by one pathway in red in the fruit fly embryo, one expressed by another pathway in blue, and in the place where they intersect, you can generate a third cell fate, and in this way achieve much more refined spatial and temporal specificity by combining these pathways in different ways. And the pathways are not combined in a hardwired way. There are many ways to combine them for different cells and different steps. Now, another important thing to remember is that these pathways do not contain the actual information on what the cell will eventually become. This is in a way like an electric current that you can connect to different devices and either activate a projector or a toaster or a light bulb. So it's the context in which the pathway is activated which will determine what the final outcome will be. It's not just the pathway. The pathway is more like a switch, but it depends what switch you trigger. So, I mean, as a paradigm, this is a street of immigrants in Somerville. There is the Hispanic barber and the Brazilian coffee shop and the Italian restaurant, and all of these people are looking at the same street, but everyone sees something different depending upon where you came from. And the cells are different each time. I mean, the same pathway can trigger the same cell at different junctions, and the outcome will be different because the cell that receives the signal has undergone modifications and changes in the course of time. And finally, another way to generate complexity in a sort of simple way is to divide the body into modules. So our body is divided into vertebra. Invertebrates divide their body into segments, and initially you just determine the number and position of the segments or the vertebra without allocating particular fates or functions to each one of these domains. It's only in the last step that you actually allocate specific features that are unique according to the position in the body. I mean, like if we look at these vertebrae, they would be different. This is from a killer whale. It would be different in the head region, in the tail region by these subtle modifications, but these are all variations on a theme that were inserted after the basic module was put in place, in a way like the facade of this hotel where every porch has 2 chairs, but you can arrange these chairs in different ways and achieve modularity by variations on a common module. Now, we talked about communication between cells, but the example I gave you initially was that of close communication. So the one cell becomes a neuron, it touches its immediate neighbors, and it inhibits them from assuming a similar fate. But in many cases in development, we actually pattern tissues across a broad field. And the term actually for this kind of patterning was coined by Alan Turing, and it's called patterning by morphogens. And the idea is that you produce a substance which is secreted from the cells. I mean, this cell layer will be the future wing of the fly, and the cells here in this region make this white protein which is secreted outside of the cell. So you secrete a protein outside the cell, and it gets dispersed to the neighboring cells, but as it gets dispersed, it gets diluted because it breaks down, it's taken up by the cells, and so on. So in essence, the concentration of this protein will tell you how far you are from the source of its production. So in this case, the cells know how to read not only the presence and absence of this protein, they can actually tell how much of this protein they see. So in a way, it's again a single word, but you can say that word loudly or softly and achieve another level of information. So in this case, a single protein patterns an entire field of cells. I mean, very much like this instructor in the museum. I mean, she talks, these people will listen, but these people hear her more strongly, and these people hear her fainter, depending upon where they sit, and the information spreads in space. So a single protein can actually allocate different kinds of information in space based upon its concentration. And the cells will develop into different tissues or sub-tissues depending upon the level of protein that they encounter. Okay, let's skip this one. Okay.
Is there also a process of repeating, retransmitting across cells? The cells get a message?
Right. I mean, so yeah. So there are possibilities of relay that you actually. There are still very big debates, even though it's such a central concept, on how do you actually distribute this morphogen. Is it done extracellularly, or is it transmitted between cells, or does one cell relay the message to another cell? There are now also indications that cells send very long processes, like neurons, to the source and take up this substance in that way. We can rule out the idea of relay by another signal by showing that the cells in this region are still sensitive to the same molecule. If it was a relay, I mean, the experiment that was done essentially was to remove the receptor in these cells. Now, if it was a relay mechanism, then these cells should be normal because, you know, the signal was relayed to them through another pathway and so on. Turns out that there are mutants, so this indicates that they're actually receiving the same original signal, but how the molecule was actually transmitted is still an open question. Okay. Now, there's a bit of a paradox in what I told you if we consider evolution of patterns. I mean, as you know, I mean, evolution is the biggest thing that happened in biology in the last 150 years or so. And Darwin encountered, or his first trigger to the idea of evolution came upon his trip with a beagle to the Galapagos Islands. These are islands that emerged from the sea between 600,000 to 4 million years ago. And yet, we can see in these very young islands new species of birds and turtles and so on. So it means that evolution of pattern is working in a very, very rapid pace. On the other hand, I told you that these pathways that drive patterning have been conserved and essentially unaltered in the last 600 million years ago. So how do you reconcile these two things? Pathways which are essentially unchangeable with rapid evolution of pattern. How do these two things take place? And in a nutshell, I mean, you can think about an instrument player. The instrument is the same, the player is the same, but if you change the notes, it can generate an infinite array of tunes and musical pieces. And in a way, evolution of pattern operates in the same way. So if we think about a gene, the structure of a gene in the genome, it's actually comprised of two basic parts. One part is the part that contains the information on how to make the protein. And this part, as I told you, is unaltered. The proteins that convey the information between cells are unaltered, or essentially unaltered in the last 600 or 800 million years. But every gene contains another part, and this is the regulatory part. It's a part that will not be manifested in the protein, but this is the part which tells the gene when to be expressed, where to be expressed, how much. Very important. I mean, essentially, this is what determines the fate of tissues and so on. So it turns out that evolution of pattern works on the regulatory sequence of cardinal genes, and preferentially at the genes that sit at the top of the hierarchy. I mean, evolution is not a guided process, it's a random mutations, but the mutations that will affect the expression of genes at the top of the hierarchy are the ones that alter pattern. Think, for example, about increasing the size of the eye of an insect. So if you take the gene that determines where the eye will form and just slightly alter its expression pattern, just make it a little bit broader or a little bit narrower, in essence, in one step, you will modify the size of the emerging eye with a single genetic change. So in a way, it's again similar to the Spearman experiment, just much less dramatic, making a subtle alteration in the regulation of a gene at the top of the hierarchy will generate a very significant difference in the morphological outcome. And this can be seen over here. These are wings of different fly species that have diverged over the last 60 million years. And every species, as you see, has a very typical pattern of pigmentation. In all cases, it's exactly the same proteins that produce the pigment in each of these species, but the position where these genes will be expressed in the developing wing is what determines the final pattern. Now, up to now, we talked about the communication between cells, essentially the instructions and decisions that cells have to make on what they will become. This is in a way like buying a plane ticket. Obviously, you have to buy the ticket, otherwise you won't go anywhere. But just buying the ticket doesn't take you to your final destination. You actually have to board the plane and get to your destination. And the same feature happens in development in the sense that there is a switch between the actual decision to the execution of that decision, to the actual formation of the tissue that this cell will make. And the execution is very different in the sense that whereas the actual decisions employ a small number of genes, the execution of fate can involve hundreds of genes in each tissue. It's a very elaborate process, and it involves many, many changes. And just to give you a flavor for what some of these changes or processes could be, so you can generate patterns in a variety or execute patterns in a variety of ways. One way is that you can generate patterns not necessarily by adding cells to a particular tissue, but actually by removing cells. I mean, like this stone maker in Rajasthan that removes pieces of stone to make this pattern. The way that our digits are formed starts from a situation where this entire limb primordium is full of cells, and the cells between the digits die. You can see it here in red. These are the cells that are undergoing active cell death. They're actually eating themselves alive, and this will generate the spacing between the digits in a very specific manner. Another important process that shapes the body pattern is cell migration. Cells can be born in one place and migrate to another, and sometimes the migration route of these cells essentially generates the structure of the organ they will form. So in a way, I mean, this is like a shepherd that leads a flock of sheep. These cells are the cells that will give rise to the breathing apparatus of the insect. So it's essentially an interconnected network of tubes that arises from patches of cells in the embryo, and these cells are now migrating in a stereotypic way, and essentially every edge here is following instructions from the neighboring tissue, very much like the sheep are following the shepherd, and these instructions essentially lead them to migrate in a particular direction and produce this elaborate structure. So execution of pattern is an important aspect, and in the end, I mean, I think one can think about embryonic development not like a mold where you fill the mold with with plaster, whatever, and make multiple identical such molds. I hope that what I showed you convinced you that embryonic development, each embryo is a new creation in a sense. It involves a new interaction between cells and so on, and therefore it's really a small wonder, or maybe a big wonder, that if you look at individual flies in this case, at the wings of individual flies, they are so similar to each other, despite the fact that each one underwent its own process of creation. So in a way, I mean, this is like not like a mold, but more like a ceramic artist that has to reproduce or to generate multiple such bowls. Each bowl is a new creation, and you have to be very precise if you want to generate them in a reproducible pattern. So how does everything that I told you relate to human health? So we started with a frog embryo and then talked about the fruit fly embryo. Is this really relevant to human health? Or can we in the future actually generate foreign body parts that, or not foreign, that actually use our own cells to generate spare parts from our own body to cure different diseases? And is the information that we gained so far relevant in this direction? I should emphasize that for me, both in the book and in the lecture and so on, it's important to put this aspect at the end of the lecture and not at the beginning of the lecture, because this is the way that things really developed. I mean, people started from basic science, from curiosity-driven research, and as it turned out, I mean, they uncovered very important medical principles. It didn't start out with people wanting to cure diseases and going out and to cure these diseases, but started the other way, and it's really important that this kind of basic research will be maintained in the future, and it's actually the way that science is funded now. This kind of funding for basic research is very much endangered, I think. So why is this relevant to human health? So you probably heard a lot about the term stem cells. I don't know, beyond the buzzword, whether you went more deeply into it. And the idea behind stem cells is that the process of cell differentiation does not end at the end of embryonic development. Actually, cell differentiation continues throughout adult life, and we have several tissues in our body that continue to renew themselves every day throughout our lives. And this includes the skin and the hair and the lining of the intestine and the hematopoietic system, the blood cells. All of these tissues are constantly generating new differentiated cells where the old cells are replaced and die. I mean, just to give you a feeling, every day we replace 250 grams of cells in our intestine. So it's a very extensive process, and every 3 days we completely replace our inner lining of the intestine. We replace our blood cells every 120 days. Very, very extensive process. Now, obviously, you need such a large pool of undifferentiated cells to carry out this process throughout our lifetime that we cannot carry these cells with us as a bag of undifferentiated cells throughout our lifetime. This will simply be too heavy, right? So what you have to do is to keep maintaining or generating undifferentiated cells, and in parallel, take some of these cells and use them to replace cells in the different tissues. So the idea behind the stem cell is the following. This is a cell that is maintained in a proliferative, undifferentiated state, but every time that it divides, the two progeny are not identical to each other. One daughter cell will maintain the features of the mother cell, namely proliferative and undifferentiated, while the other cell will go on to differentiate and form the cell types that are necessary in the tissue where it resides, be it blood cells, skin cells, or whatever. Now, obviously, this is a very critical balance because you want to this will maintain the proper number of stem cells and will also generate a steady supply of differentiated cells, but you really want to maintain that balance because if you skew it in one direction, I mean, for example, if you generate too many progeny stem cells, you'll get a tumor. If too many cells will differentiate, you will lose your stem cells and you will not be able to regenerate them. So you really have to maintain that balance very tightly. And the way in which it is maintained is by a concept which we call the niche concept. And I'm showing it here through the lens of a fish, a zebrafish. So this is the lens. And around the lens is a tissue, which I'll explain in a minute what it does, which is called the niche. This is where the stem cells reside. And the differentiated cells that arise from the stem cells are pushed outwards, and they will generate these neurons that emerge from this structure. Now, what is a niche? A niche is actually a geographical entity. It's a fixed space, and only in that space can the stem cells maintain their undifferentiated identity. How is that space generated? Because it's a space which contains many goodies, many proteins that reside only in this region and not in another region. So think of a stem cell that divides. It now generated two cells. There is no place for the two cells within this geographical niche. So one will stay, the other one will be pushed out. Once it's pushed out, it differentiates, and in this way you maintain the balance. So the actual geographical restriction of the size of the niche will maintain a fixed number of stem cells, and this also has another advantage. Since the limitation is a geographical one, you could have a situation where as a result of a division of one stem cell, its two progeny will stay in a niche, while another cell, the two progeny may differentiate. You are not that strict in terms of allocating these fates to the progeny of a single cell, and this means that if cells that divide a lot and can accumulate mutations can be pushed out of the system and can be replaced by other cells. So how do you convey this concept of a niche? Well, if you think about a bakery, bakeries have what they call the mother, which is their concoction of yeast cells that they maintain alive and keep feeding them with sugar and flour and so on. And every bakery has its very special mix of yeast, and you have to keep it alive and propagate it and so on. But in parallel, you take out of this mixture the yeast that will make your bread, but the bread is a dead end as far as these yeast are concerned. So you really have to maintain this tight balance between maintenance of your yeast population and the production of bread on the other hand. So how can we use stem cells for medicine? So ideally, what we would like to have is to be able to have stem cells that are totally undifferentiated. The stem cells that I described previously are stem cells that are specific to specific organs, to the intestine, to the skin, and so on. Ideally, you'd like to have stem cells that are completely undifferentiated. And now that we know more or less the different communication pathways that can drive cells to differentiate in different directions, we can take these cells, non-differentiated cells from our own body, and drive them to generate the tissue of interest. But how do we find these cells, or how do we generate an unlimited supply of these non-differentiated cells? So it turns out, and this is a finding that was made in 2006 by Yamanaka, for which he received the Nobel Prize, that you can actually take differentiated cells from the skin, and by expressing only 4 proteins in them that he identified, drive them all the way back to their non-differentiated state. And having done that, you can now push them in different directions. These are cells, human cells taken from the skin and then pushed on to differentiate as neuronal cells. These are typical structures that the neuronal cells make in culture. In a way, you can think about it like people who have lost their job and are now trained in a totally new direction that has nothing to do with their previous expertise and occupation. Now, this is not a trivial undertaking, because in order to push cells back to their non-differentiated state, you have to erase not only the genes that they expressed in that particular tissue, but also epigenetic changes, which is what you asked at the beginning. What are epigenetic changes? These are changes on the DNA that are not related to the actual base composition of the DNA, but rather are sort of auxiliary changes that are superimposed on that. And they could be either methyl groups that are inserted on the DNA, or proteins that will bind particular regions and in this way maintain that region either open for expression or closed for expression. So it's a whole variety of changes that are superimposed on the crude genetic information, which are very important and difficult to erase. And if you look at this tattoo, I mean, you can sort of envisage how difficult it is to erase such epigenetic changes in order to come back to the ground state, which will now allow you to differentiate in different directions. So there's actually a lot of hope in using these findings and the identification of stem cells and induced pluripotent stem cells for medicine. In tissues which are working in suspension, like the blood system, this information is already being used extensively to take cells from the peripheral blood and push them back to a less differentiated state and so on, because there the tissue architecture is not important. It's just a matter of adding the right goodies and pushing the cells in different directions. This, of course, becomes much more complicated when we deal with tissues which have a very precise three-dimensional organization. For example, here we're looking at the Isle of Langerhans. This is a cluster of cells. We have about 1 million of those in our pancreas, and these are the cells that produce the insulin, and these are the cells that die when we have when people have juvenile diabetes. And you can see the insulin granules here in these white little granules that the cells will secrete. So if you want to regenerate such an organ, which is comprised of 5 different cell types with very intricate relations between them, with a very typical three-dimensional structure, it of course becomes much more complicated than just adding the right goodies. You need a scaffold. There's a very specific order that you have to maintain. But still, I mean, we are on the right direction, and I predict that in the coming years for different tissues, we will have the capacity to generate spare parts from our own cells, meaning that one will overcome immunological rejection that is currently a problem in organ transplant, and be able to really harness our basic understanding of biological research and embryonic development for actual therapy and generation of spare parts. So just to conclude, so I hope I convinced you that cell fates during embryonic development are not a predetermined fate, but rather it is the interaction between cells that drive embryonic patterns. Every cell has multiple options, but talks to the neighbors, and this will determine the final outcome. So you need communication on the one hand, and you need rules. And the rules are embedded in the genome. This is why the DNA dictates the final pattern, because it contains the rules. It contains the information on how to make these signaling pathways. And the combination will generate patterns. Communication between cells is a universal feature. All cells, I mean cells in all multicellular organisms, use the same communication pathways, the same principles to generate the different organs. Despite the fact that the organs may look very different in the different species, they actually are formed by the same principles and rules. The formation of pattern is a very dynamic aspect from an evolutionary standpoint, and one can generate enormous variations in patterns very quickly over evolutionary timescales simply by altering the expression pattern of genes that sit at the top of the hierarchy of interactions between cells. And finally, stem cells, which are undifferentiated cells in our adult body, provide the hope for many promising directions of therapy. So just to conclude, I would like to thank the Radcliffe Institute, which really provided a wonderful environment to initiate and carry out this project, the Sloan Foundation, which provided some of the support for publishing the book and the website, my lab for ideas and images, colleagues for providing many images, and my wife for continuous input and criticism, and so on. So now, I'll stop, and I'll be happy to take questions or to relate to the more deeper questions that were asked during the talk. So epigenetics, we sort of answered that. I can elaborate more if you want. There was another question during the talk. Yeah.
Is stem cell and differentiated a relative concept? I mean, you say stem cells which are at the very top, and then there are stem cells which can only do certain.
So there are different types of stem cells within. The normal types of stem cells are already sort of semi-differentiated. So you can be a stem cell of the skin, or a stem cell of the intestine, or the hair, and so on. So it's a you're already slightly differentiated. You're not completely naive. What one achieves with these induced pluripotent stem cells is really to be able to take fully differentiated cells back to really their most primordial state, and in this way, first of all, you have an unlimited supply of cells, and you have many more options in terms of where you want to push them. Yeah. Yes.
Fully developed cells do not multiply at all, or some of them multiply?
Pardon?
Fully developed.
Yeah.
Yeah. Not multiply.
Yeah. Usually they don't. No. So, for example, in the liver, if we have an injury in the liver, many times the way that the liver compensates is not by generating more cells that will divide, but simply by having the existing cells grow in number. Actually, they double their DNA content, but they don't divide. You get larger cells that fill in for the cells that are missing. In some cases, there are claims that tissues can repair themselves by taking essentially to do normally what the induced pluripotent stem cell experiment did, to take differentiated cells, push them back to their non-differentiated state, and allow them now to proliferate and differentiate and so on. But this is only upon injury and only in relatively rare cases. Yeah.
Can I ask you?
Yeah.
You talk quite a lot about these rules and communication together. And can you comment on what is the difference, if any, when you are talking about this patterning of body patterning and, let's say, a connectome of the brain? I mean, the connection that are done during our life, basically coming back and forth and so on. So what is the commonality and where the big differences are all about?
Mm-hmm.
I mean, building a network of quite a fascinating pattern in the brain throughout life.
Yeah. Okay, big question, huh? Yeah.
Big one.
I guess one can think about the brain also, broadly speaking, in two levels. One is how you actually form this connectivity, and also another level, and I talk a lot with my neighbor Amos Arieli about it, is how once you've actually built the structure of the brain, how do you, when you look at an image from the world, how do you incorporate all that information in order to understand what it actually means? And at least, from our dialogues, at least with respect to interpreting visual information by the brain, there are lots of similarities in the sense that there are so many possibilities that you cannot a priori predict all the possibilities. Just like in embryonic development. If we have billions of cells in our body, we cannot a priori tell each cell what it will become. Just too complicated, too many cells, and so on. So you have to build on rules that somehow take the information from the general to the specific, and this combination somehow generates an understanding. So I think in many ways it will have to be, with the brain as well, about rules and communication that somehow build a more elaborate structure. It's not, you cannot have a blueprint for the final structure from the outset. It's just too complicated. And it's not, another issue about a fixed blueprint is that it's not a robust pattern in the sense that it's very sensitive to changes and fluctuations. I mean, suppose one cell dies. If this is the cell that had to be that organ and it dies, I mean, who will replace it? But in a dynamic network, if every cell can be everything, then it's much more plastic, much more flexible, and so on. Yeah. Okay, so thank you.
So thank you. Just before taking off, we would like two things. First, to give you a small token of our appreciation. So also you can read about us and about the Institute and enjoy this one. And actually, there are some of this book that we can sell. I'm not getting any percentage. So it's 110 shekels. So whoever wants this presentation in a book, just come to me and you can have it. I don't know with or without the signature of the author, but anyway.
It's cheaper with a signature.
Yeah, yeah. Yeah, exactly. And thank you so much, Benny.